Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A level Biology Unit 1 potential questions. Let's begin with the first part. Topic 1, Molecules, Transport and Health. Here they will ask you about the chemistry of water. You need to know the shape as well as the bond angle of water molecules. You need to know that water forms hydrogen bonds between their molecules, and the bond angle between molecules is 180, while the bond angle within a water molecule is 104.5. You need to know that water is used as a solvent, and the details of how water acts as a solvent in dissolving ionic compounds like sodium chloride. You need to learn about other uses of water as indicated on page 7 of your textbook. When we go to carbohydrates, remember carbohydrates can be monosaccharides, which include trioses that are 3 carbon, pentoses that are 5 carbons, and hexoses that are 6 carbons. You need to know that glucose, fructose, as well as galactose are also monosaccharides. These are hexoses. You need to know that maltose, sucrose, and lactose are disaccharides, and you need to know what they are, meaning maltose is a mixture of glucose and glucose, sucrose is glucose plus fructose, and lactose is glucose plus galactose. You need to know the differences between the two pentoses, which are ribose as well as deoxyribose. You need to know that a glycosidic bond can form between 2 glucose molecules, or you could say glycosidic bonds form between glucose molecules, and these glycosidic bonds could be 1,4 or 1,6, depending on how the alignment of the glucose molecules is. You need to learn how to draw at least 2 glucose molecules and show 1,4 glycosidic bonds and at least 2 glucose molecules and be able to show the 1,6 glycosidic bonds. You need to learn to do the testing for reducing sugars using Benedict solution. And you need to know the observed colors that happen during this experiment. You need to learn about polysaccharides like starch and glycogen, including amylose and amylopectin. Remember those shapes in starch as well as glycogen. You also need to know to compare and contrast the structure of starch and glycogen. Going to lipids, you need to know that they can be fats, which are mainly from animals, or they could be oils, which are mainly from plants. You need to know that lipids contain fatty acids as well as glycerol, and these are linked through esterification reactions that form ester bonds. The fatty acid part can be saturated, or sometimes they can be monounsaturated or polyunsaturated. You also need to learn to draw a triglyceride. Moving on to proteins, you need to know the basic structure of an amino acid. You need to know that amino acids can be linked through condensation reactions to form peptide bonds. You need to know that a dipeptide contains 2 linked amino acids, and a polypeptide, which is a protein, contains many amino acids that could be linked up. You need to know that hydrogen bonds, disulfide bonds, as well as ionic bonds are vital in creating the 3D structure of a protein. You need to know that proteins contain primary, secondary, tertiary, as well as quaternary structures, because these could be examined. They can ask you to describe how these structures are and what type of bonds exist between them. You need to learn the details of collagen formation. Here we include the procollagen triple helix, the shortened collagen molecule, the collagen fibers, as well as the collagen fibers that are produced. On this note, you need to know that the primary structure of collagen is made up of repeating sequences of mainly glycine and often 2 other amino acids like proline and hydroxyproline. This is also common. This is an example of a fibrous protein, and this idea can be applied to any fibrous protein that you can be asked about in the exam. You need to learn that for globular proteins like hemoglobin, they contain multiple tertiary structures of polypeptides. These could be 3 or more tertiary structures that are arranged in a specific way to produce the quaternary structure. You need to remember about conjugation of proteins by adding prosthetic groups, like lipoproteins when lipids are added, as well as glycoproteins when carbohydrate groups are added. We also need to remember about testing for proteins using Burette test and all the observed colors that are going to be seen. When we go to circulation, we need to know the features of a mass transport system. We need to talk about circulatory systems in terms of single and double circulation. We need to know the components of blood, like plasma, the red blood cells, white blood cells, as well as the platelets. and we need to know the functions of plasma as a transport medium. We need to know the adaptations of red blood cells to perform their function. Examples like why don't they have a nucleus? Why do they have a specific shape? You need to know the reasons for that. You need to compare and contrast white blood cells as well as red blood cells in terms of their structure and function. Continuing with circulation, we need to know the process of blood clotting, including the roles of thromboplastin, prothrombin, thrombin, fibrin, as well as fibrinogen. We need to explain why thromboplastin is contained in the platelets. You need to know about the oxygen dissociation curve and the Bohr effect. You need to know why the dissociation curve of fetal hemoglobin as well as myoglobin are on the left of the adult hemoglobin curve. That is because they have a higher affinity for oxygen at the same partial pressure in comparison to that of hemoglobin. We need to know about the structure of arteries, veins, and capillaries, and how these are adapted to perform their function. An example, why do veins have valves? Why do arteries have smaller lumens? You need to know why those are the way they are. You need to learn how to fully label the heart. You do not need to draw, but you need to know how to label. You need to carry out description of double circulation in humans, explain and give a detailed explanation as well as a description of everything that goes on. We need to know the details of the cardiac cycle, like atriosystole, ventricular systole, as well as cardiac diastole. We need to know the details of progression of atherosclerosis. This is common, and you need to know the detailed explanation as well as the description. We need to talk about atherosclerosis and aneurysms. We need to know about atherosclerosis, blood clots, and heart attacks or strokes. Going on to cardiovascular diseases, we need to know the definition of a risk. We need to be able to tell apart a correlation as well as a causation. Here we also need to know the non-lifestyle risk factors for CVD, like genetics, age, as well as gender. We also need to know lifestyle risk factors like smoking, inactivity, high blood pressure, obesity, high cholesterol, and so on. And how these affect atherosclerosis and how that can lead to cardiovascular diseases. We need to learn to practice explaining and evaluating data from past papers on these factors. I'm talking about the risk factors because many times they bring questions as well as data on these points. They usually bring 6-mark questions where they ask you to explain, to describe, or suggest. In these questions, you need to write coherently and you need to use keywords. Sometimes they can ask you questions to explain deeper. You need to write what you get from the data that is presented to you. You also need to add some extra information from your own knowledge because sometimes that is awarded an extra mark. We also need to know how these risk factors can be tackled, like how obesity, smoking, inactivity, and so on can be minimized. You need to learn about BMI calculations and explanations relating to data given to you, as well as using waist to hip ratio in calculations. You need to learn to interpret data on antioxidants and CVD. This is common, and sometimes they can ask you to carry out tests for vitamin C in a food sample using DCPIP. This is explained in detail in your textbook on page 64, so please visit that and try to ensure you fully understand. You need to learn about controlling blood pressure using antihypertensives, and on this note, they could ask about diuretics, beta blockers, sympathetic nerve inhibitors, and so on. You also need to learn how to control cholesterol using statins, stenols, as well as sterols. And lastly, controlling blood clots using anticoagulants like warfarin as well as inhibitors like aspirin. From topic 2, membranes and transport. The first part is the structure of the cell membrane. Here you need to learn how to draw and label the phospholipid. And on that note, you need to remember that a phospholipid has only 2 tails. You need to clearly understand the meaning of hydrophobic as well as hydrophilic. And you need to know that the cell membrane is a bilayer of phospholipids. You need to know the structure of the cell membrane, that it contains phospholipids, it contains proteins, carbohydrates, cholesterol, and so on. Moving on to transport across the membrane, you need to know how to describe the passive transport mechanisms across the membrane, like diffusion, facilitated diffusion, as well as osmosis. You need to know the description of active transport mechanisms, across the membranes. These include active transport through the proteins, endocytosis as well as exocytosis. You need to know osmosis in living cells, including plant and animal cells, as well as experiments to show the effects of placing cells in solutions of different salt concentration. This includes plant and animal cells. And here they can ask about plasmolysis, flaccidity, as well as turgidity. Lastly, on this, you need to know about osmotic concentrations in solution, like hypertonic, isotonic, and hypotonic solutions. Moving on to gas exchange, you need to know the properties for gaseous exchange systems based on fixed law of diffusion. Properties like a large surface area to volume ratio, greater concentration gradient, the shorter diffusion distance or the thickness of the membrane. If it's thin, then there is going to be greater diffusion. We need to know the components of the human gas exchange system in terms of their functions and adaptations. We need to know the adaptations of the alveoli for gas exchange. Why are they very efficient at carrying out this function? You need to know the adaptations they have. We need to know the detailed description of inhalation and exhalation, including what changes are going to occur on the intercostal muscles, the diaphragm, the pressure, as well as the volume changes that occur in the chest cavity, and so on. Next, when we go to enzymes, they will ask you about enzymes and their relation in metabolism. This includes catabolism, which is going to be breaking, and anabolism, which is building. They can ask you how enzymes work. You need to know in detail the lock and key model as well as the induced fit model. They can ask about the effects of temperature on enzyme activity, including graphs interpretation as well as Q10 calculation. You need to know that. They could ask the effect of pH on enzyme activity or on the rate of reaction. You need to know that as well. Sometimes data is going to be given to you and they want you to use graphs or tables to use that to answer specific questions. They could ask about DNA and RNA, and here they could ask about mononucleotides versus polynucleotides. Here you need to know the components as well as the bonds present in the mononucleotides as well as the polynucleotides. For example, you need to know that a phosphodiester bond is formed between the phosphate of one mononucleotide and the sugar of another mononucleotide. You need to know that purines include adenine and guanine, while pyrimidines include cytosine, thymine, and uracil. You need to understand fully about the conservative and the semi-conservative models in detail. This question has come twice, including the isotopes used. Remember, they use nitrogen, 14 as well as 15. You need to understand the details of DNA replication, including the functions of DNA polymerase, like lining up the nucleotides, the function of DNA helicase for unzipping the DNA, as well as the function of DNA ligase for the formation of the phosphodiester bond. You need to remember the details that the genetic code is read in codons or in triplets or in threes. You need to know that the code is non-overlapping and it is degenerate. To be degenerate, it means that it contains more information than needed. You also need to fully understand the details of protein synthesis, transcription, the whole of it, as well as translation. When we go to gene mutations and genetics, you need to remember the details of gene mutations like substitution, deletion, as well as insertion. You need to fully understand how gene mutations can affect the phenotype. Here they could use it as examples. Examples here include cystic fibrosis, thalassemia, as well as sickle cell anemia. We need to know the definitions of what is a genotype, what is a phenotype, what does homozygous mean, heterozygous, allele, dominant, recessive, and so on. And actually, this can be given in relation to a specific question, so your definition can be twisted a little to suit the specific context. We need to practice carrying out genetic crosses, and in doing so, you also need to know the advantages of carrying out test crosses. We need to know what codominance means in the ABO blood type. Like somebody, they have A and B antigens, of course their blood type is going to be AB because there is codominance. You need to know how to interpret pedigree diagrams. Here you will have to use a lot of past paper questions to practice. And you need to know about sex linkage and disease in humans, including the red-green color blindness and hemophilia. We need to know about cystic fibrosis, the details of what genetic changes occur and how they lead to this condition, And we need to know how cystic fibrosis affects the respiratory system, the digestive system, the reproductive system, and the sweat glands of people who have this condition. For example, why do they produce sweat that is so salty? You need to know why. Lastly, we need to talk about genetic screening. Of course, they're going to ask you about prenatal or pre-implantation genetic screening. We need to know why prenatal genetic screening like amniocentesis and cryonic villus sampling are carried out. You need to look in your textbook on page 143 and understand fully the details of that. And you need to know the benefits of this genetic screening as well as the risks involved. And for pre-implantation genetic screening, again, we need to know why it's carried out, the details of the procedure, as well as the advantages and disadvantages of this method. This brings me to the end of the potential questions that I thought can come in Unit 1. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.